Okay, so we're going to start with glycolysis. Glycolysis is basically just a whole cycle to, the goal of it is to break down this 6-carbon glucose down into 2 3-carbon molecules called pyruvate. It occurs in the cytosol, and the rate-limiting enzyme is phosphofructokinase. Basically, that just limits the rate and the speed of this whole reaction and the rate that the ATP is being produced. So if the ATP levels are high, the level or the activity of PFK is going to be low if ATP is high. But on the other hand, if ATP levels are low, then PFK is going to be more active. So we start with the 6-carbon glucose molecule. You can also start with glycogen, but for this example, we're using glucose. We're using glucose. Basically, you start out by adding 2 phosphate molecules to the first carbon and the carbon 6. Then you go down here, and that molecule is converted to pyruvate, while it converts, like during all of this, it converts the NAD plus to NADH, while also converting 2 ADP molecules to 2 ATP molecules. So you have a net yield of ATP, you get 2 ATPs. So then we move to the Krebs cycle. With this one, you take the pyruvate from the glycolysis cycle that we did previously. The pyruvate molecule is converted to acetyl-CoA. Carbon dioxide and an NADH are both released. This looks kind of confusing. I first drew it this way, a little bit more simple, a little bit more of a visual, but then I added more detail to the outside involving like the enzymes and things like that. So here you're starting out, like I said, with the acetyl-CoA. You go from a 6-carbon molecule down to 5, 4, yada, yada, yada. And then basically you have all of these coenzymes coming off the NADH, and the CO2 is being released. You have right here the ADP being converted to ATP. And then you also have FAD converted to FADH2. The rate-limiting enzyme here is isocitrate dehydrogenase. And another thing to note, the reason that it's called I refer to it as the Krebs cycle, but it can also be called the citric acid cycle. It does, here at the beginning of the reaction, start with citrate, otherwise known as citric acid. So that's converted into isocitrate, then alpha ketoglutarate, down to succinyl-CoA, succinate, fumarate, malate, oxaloacetate, and then the cycle can continue. One thing to note is that you have to go through the cycle 2 times, 2 loops per glucose molecule. So we started glycolysis with 1 glucose molecule. With that 1 glucose molecule, we have to do the Krebs cycle, the loop, 2 times. So the Krebs cycle, the yield, we're going to get 2 ATP because you go around 2 different times. 6 NADH, 2 FADH2, and 4 carbon dioxides. And one other thing to note that I did make a note of down here, it's important to pay attention to everywhere that this is occurring, where you're taking NADH plus and converting it to NADH, or FAD2 plus and converting it to FADH2, because that is where oxidation is occurring. And then one last thing occurs, the Krebs cycle occurs in the mitochondria. So finally, we are at the electron transport chain. I am not an artist, obviously. This kind of looks like a mess, but I'll break it down. This occurs in the mitochondrial membrane. You can see that this is the outside of the cell, this is the inside of the cell, and this weird looking thing right here is the membrane. So all of this is kind of occurring down right through here. So basically we're taking these NADH molecules. I'm sorry, let me start here. You can see all of these giant circles right here. Those are the electron carrier proteins. So that's what is carrying all of this down the gradient through the membrane and whatnot. So you're starting with the NADH molecules. These are your coenzymes. Those are basically taking the electrons. The electrons are going into the electron transport chain. The protons, or H pluses, are going out of the cell. So these are what we want. So these are going all the way down here, and simultaneously, while this is all happening, you're having NAD go out as well. But all the electrons are going all the way down here. And when it gets down here to the final electron acceptor, which is oxygen, this is where oxygen comes in. Oxygen is the final electron acceptor. The electrons are coming out, pairing with some of the protons, or the water, sorry, not the water, the H pluses, and forming also with some oxygen, water. So basically, after all of that, you're getting water. Also, the H pluses are coming through here. This is ATP synthase, and you have ADP coming through as well. All of that is coming together to form ATP. This is where all of your ATP is coming from this whole thing. So your rate-limiting enzyme is cytochrome oxidase. The net yield from just the electron transport chain is 28 ATP. This is based on like how many NADHs that you had, or FADHs. And also remember, this could be slightly different based on whether we started with glucose or glycogen at the very beginning of glycolysis. So, but anyways, for this example, we get to 28 ATP. And the total yield for glycolysis, Krebs cycle, electron transport chain, the whole thing is 32 ATP from that one molecule of glucose that we started with.